Welcome to Nepal, home to the majestic Himalayas where the beauty of nature meets incredible biodiversity. Nestled in this unique landscape are ecosystems rich in flora and fauna, some found nowhere else on Earth. Discover Nepal's wonders, where every peak holds a promise of discovery. In this treasure trove of life, groundbreaking biotech research is thriving at Research Institute for Bioscience and Biotechnology, unlocking the secrets of nature to develop sustainable solutions. From enhancing agriculture to preserving biodiversity, Nepal is at the forefront of innovation, merging traditional knowledge with modern science. Join us in exploring the Himalayan lactic acid bacteria project with Prajwal Rajbandhari, President of Research Institute for Bioscience and Biotechnology, Nepal.
I'm Sajita Kanun from microbiaX and your host for today. A very warm welcome to you, Prajwal Rajbandhari, and we are happy to have you as the esteemed speaker today. This session is for International Microorganism Day, and we are privileged to have you as a guest speaker. And Raj Bandhari he's a renowned expert in biotechnology, and he has more than a decade of experience in this sector. He's also the co-founder and president of a Research Institute for Bioscience and Biotechnology, which is also called as RIBB Nepal. So I would also like to welcome all the participants. So without further ado, I'm giving over the stage to Raj Bandhari. So you can start.
So thank you, Sajita. So first of all, I would like to congratulate everyone and for International Microbiology Day. And also I would like to thank Sajita and microbiaX for giving us this opportunity to share our research story regarding Himalayan lactic bacteria project. So today, I'll be before going to the Himalayan lactic bacteria project, I'll be talking about our institute, a small brief introduction about our activities that we do in our lab, and then I'll go to the Himalayan lactic bacteria project. So as Sajita mentioned, I'm Prajwal, and I lead this research institute. I lead this institute, Research Institute for Bioscience and Biotechnology. So regarding RIBB, so I finished my master's from Kafir University back in 2010, and then I, along with my friends and also from professor from Kafir University, we came up with the idea of co-founding a research institute that works on biotechnology area. So in 2011, July 4th, we co-founded a research institute. So it's already been 13 years. And we have like different areas of research uh, in our institute. So before going to all this research activity, so first of all, I would like to uh, tell about our research campus. So it's been like more than 3 years, uh, 4 institutions. On my left, there's Carver Research Institute for Biological Sciences. We have Photon Research Institute, and we have Research Institute for Bioscience and Biotechnology, and a company called Crimson Tea. So 4 different institutes came together 3 years before to make co-found a research campus in Nepal. So our research campus name is Biomedical Research Campus, and it's a hub for intelligent research in Nepal. And if you see inside our campus, we believe on collective intelligence. So we bring intelligence from all the institute, from all the researchers, and to conduct a research in different field of sciences. So so over here we have like 3 objectives. One objective is to foster collaboration between research institute and companies in Nepal. Second one, to stop instrument duplication and also to stop research duplication in Nepal. And fourth one, so nowadays, we don't live in silos. So we believe if we work together with different fields like biology working with engineers, engineer working with doctors, then that will give a very good output in coming years. So we believe in interdisciplinary research. So these are different 3 motto of our research campus. So it's already been more than 3 years. So inside our campus, we also established a biobank. Probably this is the first biobank in Nepal established in research institutes. So inside this biobank, we have like all together more than 2,000 human samples. This human samples comes from plasma, from PPMC and cell lines. So these are all stored in our biobank. in order to conduct different research in HIV, cancer, and other biomedical applications. So this is a small glimpse of biotechnology lab that is owned by Research Institute for Bioscience and Biotechnology inside Biomedical Research Campus. So we have 6 labs. On my left, on my top left, you can see the microbiology lab over here. Undergraduate student and master student are doing their research. On my top right, we have a BSL2 lab where we do a lot of cell culturing, and also we have biobank on bottom left. So again, here's a hood for cell culture. On bottom right, so we also have a lab for natural product chemistry and plant science. So these are all different labs inside our campus that deals with biotechnology research. And this research campus and also the RIBB also give a lot of opportunity to undergraduate students and also to the graduate students. So over here you can see we also provide a lot of job opportunities. So in last 13 years we provided more than 50 research jobs to undergraduate students and master students. Beside that, we also provide thesis support to undergraduate and master students, and also we provide internship to international students and also to national students coming from different colleges and university inside Nepal. So beside research, we also engage with public engagement activities in different local communities. Over here, like in 2019, we signed an MOU between Engage Nepal with Science, a UK-based charity, uh, uh, and with RIB. So within last 5 years, we have engaged like more than 40 schools in different region of Nepal. And then our scientists over here, researchers over here, undergraduate students, they go to the community, they go to local schools, and they give different kind of hands-on activities in biology, chemistry, physics, parasitology, and also gives them some knowledge on hands-on practice on how to make like art of making different bacteria, viruses, and protozoa. So these are also different kind of activities we do beside research in our campus, in our institute. And if you see our research activities, we believe in collaborating with nature. So our researchers and also scientists from our lab, students from our lab, they go to different areas, like different zones, different geographical regions in Nepal, from hills to mountains to lowlands. So on my left, you can see our researcher named Kusam Marjan is busy with isolating, culturing lactic acid bacteria or like microbes from the milk, from the cow milk from lowlands. Over here, you can see our researchers busy on isolating some samples, like taking some samples for isolating Streptomyces from swell sample. Sorry, and on my right, you can see it's a yeast cake, which is used to make bread or making like alcohols. So we also isolate different kind of yeast from indigenous yeast cake. And over here on my uh, on lower right, you can see like how do we store samples in high altitude and also like trekking to. So so in a year we go like 2-3 treks for sampling from milk sample to soil sample to yeast cakes. So now I want to talk a bit about our institute, Research Institute for Bioscience and Biotechnology. So we believe like we need to explore the rich biodiversity we have in our country. So looking at our biodiversity, we focus 4 areas uh, of research in our institute. So first one, we focus on extracting and characterizing different bioactive natural compounds from plants, from medicine plants, and also from microbes from different niche area. Second one, Nepal is very diverse in food and also diverse in different ethnic group and languages. So we also work on traditional fermented food. Over here, we isolate different lactic acid bacteria from different traditional fermented food produced by different ethnic group in Nepal. And also, we are planning to see how these lactic acid bacteria found in fermented food is related, or that there's a correlation with gut microbiome or gut health or not. Third one over here, we also work on climate change. So it's been more than 2 years. We're working on a rice variety called Jumri Marsi, which is grown in the highest altitude in the world, like it's around 3,000 meters. So right now we're looking at different climate change parameter, like let's say carbon dioxide, humidity, temperature is affecting if there is any effect on indigenous rice variety or not. And fourth one is recently we started working on human diseases. Recently we're working on hunter-gatherers, on oral microbial hunter-gatherers, and then see correlate with their gut health and and also with their activities. And also we're looking forward to different biomedical research um, in coming years. So these are different four areas where we do research in our institute. So now I'll be talking about Himalayan lactic acid bacteria project. So on my left, you can see so how old age woman in Nepal is like making a traditional fermented food named gundruk, and she's about to dry, cut in small pieces and dry. So this is how the gundruk is made and dried over here. On my right, you can see like we have different diversity in our gut microbiome, and so we want to see the relation between how this fermented food in Nepal is correlated with gut in our native population. So as I mentioned before, so on my left you can see different fermented food from Nepal. So there is Gundruk, Mosiora, Kinema, Kalpi, Tongba, Emery, and we want to again I mentioned before like we want to see the relation between fermented food, gut health, and chronic disease in coming years. Right now we just started isolating different bacteria from fermented food. So first of all, I want to talk about. So today I'm talking about few fermented food from Nepal. You can also find this kind of fermented food like named Gundruk in India also, and also in Bhutan. So you can see there's a farm over here. So in Nepali we say rayo leaves. So usually they take rayo leaves from the farm or from their own garden, and they keep in the sun for sun drying for almost like 2 3 days. And when these green leaves converted into a pale yellow color, they keep inside a jar. It could be like a glass bottle, or it could be a plastic bottle, and they compress inside the glass bottle, and then they keep in the sun for like 8 to 9 days. So over here you can see a lot of anaerobic reaction goes on, and then uh, the lac the carbohydrate present in these leaves is converted into lactic acid. So you can see after like almost like 19 days, so these green leaves is converted into the gundruk, so it's a fermented product. So and this gundruk can be eaten like by making a soup or curry. Also, you can keep it in like without giving heat, like making a pickle of the gundruk, and it's very famous. And also, people often use it in Nepalese dinner and lunchtime. So in 2011, I went for a trek to Khopra, where you can see like a Gurung community over here. They also make a gundruk. So so gundruk is made from lowlands from 80 meters to highlands like around 3,000 to 4,000 meters, and people eat everywhere in Nepal. And second food is horticulture. So as I mentioned, Gundruk is found in different landscape in Nepal, from lowlands to hill regions to mountains. But this horticulture is only made in the lowlands from east to west in Nepal. And so these are the leaves of Chenopodium. So in Nepali, we say Betua ko sag. So in scientific name, it is Chenopodium album. And these leaves are first of all harvested, and then they are dried for like 1 day, and then you make a paste of black melting paste, and in the black melting paste you dip a few leaves over here, and then you make this kind of sticks after drying. So maybe like you can dry for 3 4 days, and then you make this kind of sticks, and these sticks uh, can be used, or are used in Nepal by making a curry, and then you take with the rice. But you can also make different kind of structures, like over here you can see circular kind of structures. So different community make different kind of structures and different fermented food um, from this uh, uh, Chenopodium leaves. So in these two projects, in the Gundru project and in this Oricon project, we are we are we are about to find we are eager to find an answer on this research question. Does production process? It's because in Nepal we have a lot of ethnic groups. So So we are planning like does production process and also like geographical location affects microbial community diversity and overall quality of the product. So we want to check is there any difference between product quality if we take from different ethnic group, and if there is any microbial diversity if we take fermented food like a gundruk agriculture from different geographical regions. So we want to see is there any effect of altitude or not. Also, we want to see if there's an effect of production process, so that there's a difference in the microbial community. So this is one research question we want to find answer. And second over here, um, maybe in I don't know in India, but in Nepal, we quite often we make this masiura. In Nepal, we say masiura. So you have like over here, I forgot, yeah, ground or beans. So these are like vegetables. So you can also make a sweet from these vegetables. But if you want to make a masiura, so you make a black lentil paste and you cut into pieces and then you mix with the black lentil paste and then you make this kind of balls over here. So this is one place in rural area in Nepal, in lowlands. So they keep on their ceiling on the rooftop uh, for sun drying. So this one is on my bottom right. So this is how we make in my house. So like in October, November, so there's a lot of sun and there's a bright sky. So in this season, we make these kinds of fermented food and then we do sun drying. So if you see like Masura and if you see Adikanjan, so they are made from different leaves and vegetables and kept together in black lentil paste and you keep for sun drying. So it's like aerobic kind of fermentation going on. But if you see on see on Gundruk, so it's more anaerobic condition. So you keep in the jar and then you ferment. So regarding Masura, so what we are thinking is like you can't culture all the bacteria present in the food because we don't have such kind of media system so that we can grow every microorganism we found in the earth. So now we are checking like is there any difference or is there any microbial community difference between uh, culture independent method and culture dependent method. So we want to see like a isolating bacteria from classical microbial method and comparing with the metagenomic tools that we are having right now. So we want to compare is there any difference between culture dependent or culture independent method. So this is the second research question. We want to find the answer uh, for our fermented food. And beside fermented fruit, we also isolated different lactic acid bacteria from milk of different cattle or different livestock found in different altitude of Nepal. So on my left, you can see we went to Khopra Trek in March 2021, where we took different samples from this Himalayan yak over here. You can see so it's a domestic yak. On bottom left, you can see again the Himalayan yak over here. So it's from Langtang region, it's from Khopra region. So there are different two regions over here. And people in this mountain region, they make um, um, this kind of fermented product like in Nepali we say churpi, and in English people often say like hard cheese, and this one is over here the soft cheese. So they make a lot of cheese in order to like save in order to save their milk from spoiling. So they convert this milk into different kind of fermented product. So this one I just keep one example of hard cheese over here. So, and if you see like there's very little knowledge on how the impact of altitude on gut microbiome. So we have not conducted this research now, but we are planning to see the relation between altitude and gut microbiome in coming years. So this one sampling area we went 3 times like to sample different milk from yak milk. Like we went to Copra in 2021, we went to Langtang in 2023, and again in Langtang in 2024. So beside hilly region, we also go to beside mountain region, we also go to hilly regions. So on my left, top left, you can see So this is the rice variety, as I mentioned in my previous slide. So this the rice variety called Jumli Marsi, and these rice are very red in color. The grains are very red in color, and this rice variety is grown in the highest altitude in the whole world, it's around 3,000 meters. So we went there to sample seeds, sample soil, also to sample different fertilizer used for growing this rice variety. So to conduct research on the effect of climate change on this native rice variety. So in the same time, in the meantime, we also collected different milk sample from goat, from cow, from Jumla. So the altitude of this region is 2,500 meters. So we also isolated different lactic bacteria from milk samples from cow milk, from goat milk, while sampling seed samples and fertilizer for Jumli Marsi. And our team also goes to different lowlands. So in Sira, similarly Chitlang, where we went to take different samples from goat, from cow. So on my right, you can see researchers are busy using the firewood nearby to culture different bacteria from milk over here. So right now, so this is a whole glimpse of our research institute, our whole research campus, owned by three research institute and one company, and also I gave a glimpse about how we're starting a research on one lactic bacteria project from different fermented food and from different livestock milk. So now a bit of result we have. So if you see, this is the Nepal map, and so we took sample from 19 district. So this project started 1 year before. So in 1 year, we collected different samples from different districts. Like we have like altogether 19 districts. So from lowlands, we have 5 districts. From hills, we have 11 districts, and from Himalayas, we have 12 districts. So these are the lowlands over here. So lowlands is symbolized by blue dots. So these are the lowland sampling site, and hilly region sampling size is mentioned by red dots over here. You can see these are the hilly regions in Nepal where we took different fermented food samples, and uh, the hilly and the mountain region is mentioned on by pointed by color like color in yellow. So you can see these are the region we went to take different fermented food samples. So from 19 districts, we took different family food samples. Among 19, so we took from different ethnic groups. So over here you can see Mughal community, you can see Tamil community, you can see Sherpa, Brahmin, Tharo, Chetri, Newar, and Gurung. So our plan is to take fermented food sample from 10 ethnic groups, and we already have like fermented food sample from 8 ethnic groups from 19 districts. So our next sampling site could be over here, like far western region from Nepal. So we haven't isolated, we haven't sampled 44 samples from this area. So our next plan is to go over here and take few samples. So this is how we are taking samples from different regions of Nepal. So if you see over here, uh, from 19 district, we took different fermented food. Like one is Gundruk, as I mentioned before. Second is Mosera, I also mentioned before, and oregano. These are the three different fermented food I also presented in my slides. But so this one is so two. I didn't have a slide for two fermented food. So one is Sinki. It is made by drying radish by cutting radish into small pieces and they dry. And the second one is they also make a radish pickle by keeping inside a bottle with oil and also some kind of spices. So these are 5 different fermented food samples we took from different districts, and these fermented food are made from different plants, from different leaves, also from the stems, also from uh, different seeds. So these are the different sources for fermented food. And if you see a map for sampling of livestock milk, so we only have like a few samples, only 4 districts. From there, we took milk samples. So two districts is from hilly region, so it is supported by red dot over here, and two is from Himalayan region. So you can see Rasua, and I forgot to mention, so maybe like from Taplis over here. So these are the reasons where we uh, isolated sample different livestock mill. So this side is like a China side, and this side and bottom side, the southern part is sorry, is very linked to India. And so we also took different milk sample from different livestock from cow, goat, buffalo, and Jumo. This Jumo is cross between yak and cow, and they produce a lot of milk in mountains region. So now in our culture collection center, in 1 year we isolated around 211 lactation bacteria from different fermented food and also from different livestock milk. So you can see like 170 food from fermented food and 37 comes from livestock milk. This is a small chat. So in coming years, what we plan is like we want to make a very good culture collection center, and that culture collection center is based on uh, different kind of beneficial bacteria we isolate from milk, from fermented food, from soil, and also we want to become Nepal first culture collection center. So our name for culture collection center is RICO. So in coming years, we want to keep more or more like lactic bacteria and also other beneficial bacteria from different sources. So a glimpse of flowchart, what we want to do in a culture collection center is like we want to build a culture collection center where there could be a lactic bacteria and could be also other beneficial bacteria, and we want to conduct research on metagenomics and metabolomics. From there, we can identify potential strains that could be useful for apply, or that could be useful for different food application and also for cosmetic application. For that, we need to improve a wild type strain. So we're also working on different collaborators in Europe, also in different companies in Europe to improve our strains. Besides that, we also want to have like different kind of native enzymes and native proteins that comes from different uh, beneficial bacteria like lactose bacteria, and also keep their genome and also do some kind of genome mining uh, in our data science lab that is we're going to build by end of this year. So this is the whole flow diagram we are thinking of for culture collection center. So this culture collection center could be also also a point for like finding different bio solution for future needs in coming days. So right now we have lactic bacteria, we have Streptomyces from different soil samples, and we do have some host plasmid and enzymes for making different kind of enzymes that is useful for PCR reaction. So, so looking at our collaborator, you see. So in last 13 years, so you can see Nepal over here. So we collaborated with two Asian countries, with Thailand and Korea over South Korea over here. And we also have like a lot of collaborators in UK, in Europe, and also in US. But our whole, if you see our research collaboration, funding and everything, that basically comes from UK and from European region. So we are very much concentrated in this region of the world. So So over here, so this is the building of our research campus. So this is the block A, and in this block A we have like three research institute. They share their facility. And in block B we have a company named Crimson Tech. So we have a two block, and 2 years back, our story was published in as impact stories in the World Academy of Sciences. Impact Story magazine. So they covered how we do interdisciplinary research, how young people, young research institutes come together for conducting different kind of research that is very much beneficial or like very much useful for a country like Nepal. And early this year, we published our story, like how we co-founded a research institute from next year. and also like what are the challenges and what are the way forward uh, uh, from Nepal to again to the whole world. Like how do we see research institute? How do we see research ecosystem as a whole in the global south? So I want to bit go through um, a summary of this paper we published in Nature. So if you see the developed country, they have a very good legal framework and they have a very good funding system so that uh, they have a very good research ecosystem. So this is like an analogy. So the flowers are like research outputs and the fruits are like innovation outputs. So if you want to grow a tree, if you want to grow fruits, then you need to have a very good uh, like supporting hands, like you need to have very good fertilizer, you need to have very good water system, you need to have very good like pillars to support these trees. So, but if you see like research ecosystem in least developed countries like Nepal, so there are a lot of red tapes, so that you can't grow uh, or your like a fruits or your like flowers in this system. So we believe so these red tapes should be removed in order to become like a developed research ecosystem in coming years. And, and the first thing we want to focus is like there should be a brain circulation and also human capital circulation between developed uh, R&D countries and also the least developed countries like Nepal. So if there is this kind of brain circulation, then it will help to grow research ecosystem in Nepal, also in South Asia. So this is the whole summary we put uh, about our research ecosystem, about our research campus in Nature paper. And if you wish to read, then you can also solve in Google for this paper. And finally, I would like to thank uh, our host, Sazita, and also her company, microbiaX, and also to all the listeners in today's webinar session. Thank you so much. So if you have any queries, so I'm there to answer your queries. Thank you.
Thank you, Prajwal, for such an insightful talk. Nepal is known for its beauty, its beautiful mountains and everything,
Mhm.
but it's also interesting to know about these microbial biodiversity and how you're tapping into it. And this is really an interesting project. So all the best wishes for your projects to develop into a good solutions for the world and for the global challenges. But I have a question from you. It's just a casual question.
Yes, sure.
So what got you into this biotech sector? Like, was it your passion or was it that, like, what got you into the sector in the first place?
Okay. So thank you for the question. So when I was studying biotechnology, no, sorry, the biology in my grade 11 or 12, I forgot, in 12, I guess. So one of my science teacher, the biology science teacher, he introduced the term like genetic cloning, gene cloning, and also like a story about Dolly. So I got fascinated with how people make Dolly and about gene cloning, about like um, how about like some knowledge about genetics. And then I thought like after completing my high school in grade 11 and grade 12, I will go for biotechnology. After finishing my high school, so in 2004, I heard like there is a course offered by Kafan University, like undergraduate program in biotechnology. So I went there and then I studied biotechnology undergrad, and then again the same university I studied um, master's in biotechnology. As I mentioned before, so I went to Korea for like 14 months to do my master's thesis. Over there, I got a lot of hands-on training on making like a hybrid kind of antibiotics from like cloning different kind of genes into the bacteria. So then I thought like let's come back to Nepal and then establish a research institute where student from different colleges, university also get training on different application of biotechnology and also get some job employment that was missing in Nepal. So this is how a small story from my side, how to get into the biotechnology.
Oh, that's interesting because you've taken a small fascination as a school student and you've developed into such a
Mhm.
big organization of research, research and development for your nation. So that's really, awesome and inspiring story. So We're so happy to have you, Prajwal.
Yes, thank you, Sajita, for your time and for your platform. Cheers.
Yeah, thank you. Thank you. And thanks to all the participants who joined us today. So thanks a lot on behalf of microbiaX. Thank you, Prajwal, once again.
Yeah.
Bye.
Bye-bye.
Bye.
microbiaX, microbe-inspired innovation.